My name is Qiangrong Liang. I'm from New York Institute of Technology. I have been attending this meeting for several years. This year, I also present my research in the meeting. And the topic of my research is in the mitochondrial quality control in the diabetic heart. So we all know mitochondria are cell power plants that provide energy for the cells, but it can also produce toxic agents like free radicals, which is toxic to the cells. And in diabetic heart, so we use reporter animals. So we found mitochondrial degradation is reduced, especially those injured mitochondria are accumulated in the heart. So we figured if we enhance the degradation of injured mitochondria, we could reduce diabetic heart injury. So that's what we found exactly. So we use gain-of-function animal models. We showed you enhance the degradation of injured mitochondria, the cardiac damage is reduced in the diabetic heart. On the other hand, if you further reduce the degradation, the damage is exacerbated. So this provides a potential therapeutic approach for preventing or treating diabetic cardiac injury. So we are doing a lot of in vivo study now, trying to use drugs to mimic the gain-of-function genetic approach. So hopefully next year, and I can provide more data to show the feasibility of this approach to reduce diabetic heart damage.